All right, so first I'd like to start taking us all the way back to some of the first attempts at chemotherapy spearheaded by Sidney Farber. And he was working with childhood leukemia. He first tried administering folic acid, which unwittingly actually accelerated the leukemia in his patients. And so that wasn't good, and they went back to the drawing board and made a few relatively subtle changes to folic acid, creating methotrexate. And methotrexate was actually able to slow leukemia and produce full remissions in childhood cancer, which hadn't been seen to that point. And methotrexate is actually still used in cancer therapy today. So that's a bit of an extreme example, but our knowledge of how drugs work in individual cancers is about, still in its relative infancy, given how complex the interactions are between drugs and individual tumors. So in the human lab, we want to take as much of the guesswork out of that calculation as possible. So we have a tumor model, which I'm representing with little circles here. And then we introduce RNAi, and these create perturbations, and we chose 8 different genes to perturb, which are commonly mutated in cancer, to try and make our results the most generalizable. So if the tumor cell here has this RNAi gene perturbation, then we have a green tumor cell. So then we take our chemotherapy drugs, for which we used over 200 of them, and we treat our multicolored tumor cells with that. And if the drug is more potent for the altered tumor cells, the green tumor cells, we'll see fewer of them after treatment. I represent that with a blue square. Now, if the alteration produces resistance to the chemotherapy drug, we see more green cells after treatment and represent that with the red square. Alternatively, it could be that the alteration doesn't do anything relative to drug resistance or sensitivity, stays neutral, and you see about the same number of green circles, and I have that as a white square. So I did this over 200 times, or for 200 different drugs, and made a red, white, and blue heat map. Now, ASLA was able to take this and render it in a much more artful manner, and that's our image in the gallery. And here, each column represents a different alteration, and each row represents a different drug. And this is just a small subset, about 1/10 of the total data. But so blue and green here represent sensitivity for the alteration towards the drug, whereas the brighter colors represent resistance. So now I'd like to turn over to ASLA to talk about what inspired her to make our image as such. I think you got to remove your mute, ASLA.
Okay. Pete's data shows cell responses to cancer therapy. In thinking about the color describing drug response, I wanted to show dark tones that represent a dramatic difference between cancer cell survival and cancer cell death. Each color code was calculated based on the interaction of the drug and the tumor. I took advantage of polar tones giving a message of hope and warmer tones giving a message of danger. Importantly, I also wanted to show black as the lack of any informative difference. In science, the lack of information is always negative, and I wanted to include this in the mood of the work. At a more detailed level, I drew small lines extending from all of the colors in the heat map. While Pete's data shows general responses of cells to cancer therapy, within every group of cells, there are significant differences. Cells are constantly evolving in response to therapy, so we need to understand general drug response as well as the pathways that cancer cells take to evolve resistance to anti-cancer agents. And this is how I try to depict the movement of evolution in this work. Thank you.
So thanks to Asa's work, we can sort of view all this as a whole and get a better idea of how to treat a patient's individual tumor, but also recognize the similarity between drugs, such as the shared redness here in this corner or the consistent blue colors in this column. So these patterns speak to similarities between drugs and how they work to kill cancer that may have been difficult to discern otherwise. And ultimately, we're able to use this to better inform which patients should receive which chemotherapy drugs. So I'd like to thank, well, first Mike Heeman for giving us the opportunity to do this research and guiding it, but also the Swanson Biotechnology Center, and particularly Glenn and the Flow Cytometry Core, the Frontier Award, which we had in collaboration with the Lippard Lab, and then the Center for Precision Medicine.